everyone, welcome back to Thinker Zone. In today's video, we are going to dive into three very important metabolic processes: the Krebs cycle, the Cori cycle, and glycogenolysis. These cycles are the central to how our body produces, recycles, and manages energy. If you are a medical, biochemistry, or biology student, this video will help you master these concepts step by step. So let's get started. As I promised in the last video, I will get back with a very informative video on Krebs cycle. So here we go. Krebs cycle. Let's start with the basic introduction. Krebs cycle, also called Citric Acid Cycle or TCA Cycle, which occurs into the mitochondria. Central pathway of energy production. Oxidizes Acetyl-CoA to carbon dioxide and generate NADH, FADH₂, and ATP. Let's break down the step one by one. First of all, we have acetyl-CoA, which combines with oxaloacetate to form citrate. Citrate re-energizes into isocitrate. Isocitrate is oxidized to alpha-ketoglutarate, releasing carbon dioxide. Alpha-ketoglutarate is converted into succinyl-CoA, releasing another carbon dioxide. Succinyl-CoA is converted into succinate, producing GTP, which is equivalent to ATP. Succinate is oxidized to fumarate. Fumarate is hydrated to malate. Finally, malate is oxidized back to oxaloacetate, ready to accept another acetyl-CoA. For each acetyl-CoA cycle, the cycle produces 3 NADH, 1 FADH₂, 1 GTP or ATP, and 2 carbon dioxides. When these reduced coenzymes go into the electron transport chain, we get about 10 ATPs per acetyl-CoA. Here's the diagrammatic representation of Krebs cycle. First of all, we will start from citrate, which converts to isocitrate, then alpha-ketoglutarate, succinyl-CoA, succinate, fumarate, malate, and then oxaloacetate is formed. Citrate is Krebs cycle starting substrate. Starting substrate we use here is citrate, while the product formed is oxaloacetate. Here's a complete description of this cycle, from where the cycle starts and how many steps are included. Here the step 1 is citrate, and we end up with the step 8, which is acetyl-CoA, which is converted to CoA. In the step number 2, NADH is converted to NADH plus H positive, and 1 carbon dioxide is released. In step 3, where alpha-ketoglutarate is converted to succinyl-CoA, carbon dioxide is added, while NAD is converted to NADH2. In step 4, GDP is converted to GTP by the release of 1 pyrophosphate, and that phosphate group goes to CoA. Succinate is converted to fumarate in the presence of FAD, which is converted to FADH2. In the next step, where fumarate is converted to malate, water is used. Then malate is converted to oxaloacetate, where NAD positive is converted to NADH plus H positive, and then oxaloacetate is formed. So here are the enzymes which are involved in the whole process: aconitase, isocitrate dehydrogenase, alpha-ketoglutarate dehydrogenase, succinate thiokinase or succinyl-CoA synthetase, succinate dehydrogenase, fumarase, malate dehydrogenase, and citrate synthase. So for the convenience of my Thinker Zone family, I have added a mnemonic here, which is, can I actually see some Filipino mothers? Where C stands for citrate, I stands for isocitrate, A stands for alpha-ketoglutarate, S stands for succinyl-CoA, S stands for succinate, F stands for fumarate, M for malate, and O for oxaloacetates. Next, let's look at the Cori cycle, also called as Lactic Acid Cycle. This pathway is important during anaerobic respiration, such as intense exercise when oxygen is limited. as I discussed in the previous video. You can also check it out if you want to understand about glucose and the concepts related to glucose like glycolysis and gluconeogenesis. In muscle cells, glucose is broken down into pyruvate, but due to lack of oxygen, pyruvate is converted to lactate. Lactate is transported to liver. In the liver, lactate is converted back to pyruvate, and then through gluconeogenesis, it is turned back into glucose. This glucose is then released into the blood and up by the muscles again. Here I have pinned a picture through which we can understand the whole process. You can see the liver and the muscle here. In the muscles, glycogen is converted to glucose, and then this glucose is converted to pyruvate by the process of glycolysis, in which one ATP is released. This pyruvate is converted by the intense exercise or any other activity into the lactate, where anaerobic process occur. Lactate is converted to lactate is then transferred to the liver, where lactate is then again converted to pyruvate, and ATP is added by the process of gluconeogenesis, and then glucose is regenerated. Here is another picture. You can also use it for the reference. Now let's move on to the glycogenolysis, the breakdown of glycogen into glucose. This process mainly happens in the liver and muscles. Here's how it works. Glycogen is broken into glucose 1-phosphate by the enzyme glycogen phosphorylase. Sorry, I forgot to move to the next slide. So let's start again. Glycogen is broken into the glucose 1-phosphate by the enzyme glycogen phosphorylase. Rebranching enzymes remove glycogen branches. Glucose 1-phosphate is converted to glucose 6-phosphate in the liver. Glucose 6-phosphate converts it into free glucose and releases it into the blood. In muscles, glucose 6-phosphate enters glycolysis directly to produce energy. Glycogenolysis is controlled by hormones, which is glucagon. Glucagon is present in the liver when blood glucose is low. Epinephrine in muscles during stress or exercise. Insulin inhibits glycogenolysis after meals when blood glucose is high. So here is a brief difference between glycogenesis and glycogenolysis. You can compare both by using these pictures. I also added the step-by-step description here. You can also use them. So here is the complete description of this topic. You can check this from where glucose is coming and from where it is going. I also mentioned the enzymes used here. You can also check this like uridyl transferase and glycogen synthetase, UDP glucose, and all the process completely explained. Thank you so much viewers for watching this video. If you want next video to be more informative, do let me know in the comment section on which topic I will make the next video. See you next time with another informative video.